Welcome to Brain Atlas, Chapter 6, Developing Brain. There are three layers of embryonic development. This is step 1, induction. Ectoderm, mesoderm, and endoderm. Ectoderm is the outer layer, and it becomes nerve tissues, and these tissues become more cells like neurons and glia. And these divide into even more subtypes like astrocytes, ependymal cells, and more. Mesoderm is the middle layer, and these tell the ectoderm cells to become nerve tissues. This is called neural induction. Endoderm is the innermost layer. So here's a map of what each of these cells become. Endoderm becomes it's the inner layer, and it becomes lung cells, thyroid cells, and digestive cells, even pancreatic cells, and alveolar cells. Mesoderm, the middle layer, becomes cardiac muscle cells, skeletal muscle cells, kidney cells, red blood cells, and smooth muscle cells. Ectoderm, which is the outer layer, becomes skin cells, neurons, and pigment cells. There's an important signaling molecule in the mesoderm called sonic hedgehog, and it is named that way because it is supposed to activate other cells. Like nerve cells nearby become specific types of glia. Nerve cells a bit farther away and exposed to medium concentrations of sonic hedgehog become motor neurons. Nerve cells that are the farthest away, which are exposed to really low concentrations of sonic hedgehog, become interneurons. So this determines what type of neuron this nerve cell will become. Proliferation. Proliferation consists of two main cells, neural stem cells and progenitor cells. Both of these divide and become many other types of cells. Mostly only two identical daughter cells are produced. Sometimes dividing asymmetrically causes one daughter cell to become a neural or glia cell and another daughter cell to continue dividing. So eventually it stops dividing after reaching adulthood, and so restoring nerve cells becomes difficult in adulthood. There's a specific time at which the daughter cell actually becomes a neuron cell or a glia cell. If this time is not correctly met, it can cause microcephaly or megalencephaly. Microcephaly is the premature switch to asymmetrical divisions caused by lack of sufficient proteins. And so this causes limited number of nerve cells and glial cells to be produced and thus can lead to infant death. This also causes a smaller brain size. Megalencephaly is this opposite. There is a post-mature switch, so it happens too late to switch from asymmetrical divisions, and so this causes excessive cell production and a larger brain size. Next is migration. This is the migration of cells that got generated in the proliferation process, and these cells migrate to their permanent places. There are three parts that bulge out during this step: forebrain, midbrain, and hindbrain. Ridges and hollow tubes are made by fusion of other ridges, and factors such as alcohol consumption, cocaine, and radiation can cause the baby inside to get exposed to these and develop intellectual disabilities or epilepsy. Radial migration. This is the specific type of there are two types of migration actually: radial migration and the standard migration. Radial migration is a specific type, and it's the migration at which the oldest neurons are in the inner layer and the younger neurons travel all the way to the outer layer to settle down there. The standard migration is where the outer neurons are on the outer layer and then the younger neurons are in the inner layer. So it makes it more convenient. Radial migration occurs in like circles, so that's why I call it radial migration. The next step is making connections. Making connections, as the name states, step involves making making connections between neuronal and glial cells. This is controlled by outside experiences such as watching TV or sitting in a math classroom. A cell body and another cell's axon terminal connect to send signals, and these determine the job what they are doing. Axons vary in length depending on the job. For example, areas where there are less axons, the axons need to be longer in length so that it can reach more distances and the signal can be efficiently transferred. Growth cone, also part of making connections, is a cone that tells the axon where to go if it needs to go forward, stop, recoil, or change direction. Only four options. These cues are given by surrounding molecules that are secreted near the growth cones, so the growth cones know exactly where they are. And growth cones are located at the tip of the axons. They come in three types: netrin, epherin, and semaphorin. Netrin was first found in a worm. So next is synapse formation. This is the space between the cell's dendrite and another cell's axon terminal. Neurotransmitters are sent through which carry the electrical signals called an action potential when the signals go through neurons. And the presynaptic neuron, as we talked about in our previous videos, presynaptic neuron is the neuron that sends off the signal. The postsynaptic neuron is the one that receives the signal. Next is myelination. This is the wrapping of axons with myelin sheath, which is produced by oligodendrocytes, which is a type of glia cell. Spaces between these chunks of myelin are nodes of Ranvier. They're called nodes of Ranvier, which allows for electrical signal to go through faster by jumping from one myelin chunk to another myelin chunk. This jumping is called a saltatory conduction. Saltatory meaning jumping. Powering back. Powering back is the process by which cells are paired back to create an efficient system. Apoptosis causes programmed cell death, so most cells die before adulthood. Trophic factors are crucial for cells to live, and if not enough of these are received, apoptosis occurs. So, one type of trophic factor is nerve growth factor, which is a crucial trophic factor for sensory neurons. Pruning, also known as neuronal plasticity, to make efficient connections and clean up unneeded ones is its job. So this is all for chapter 6. Thank you.